Hello, in this last video about the technology we use to learn about the brain, we're going to talk about technology that lets us see the activity of brain tissue. So, different ways you can do this. You can actually go in and electrically stimulate different parts of the brain, or you can record activity. So one example of electrical stimulation, I did this for my master's. We go in, drill into little rat brains and then stimulate different parts and then look at the different activity. So in the 50s, scientists stimulated a part of the brain called the medial forebrain bundle, which they called at the time the pleasure center. And if you train a rat to press the bar every time the MFB is stimulated, they'll press that bar all day long. Then they did it to humans, and humans could push a button and it would electrically stimulate their pleasure center. And we'll talk about that later in the semester. That'll be a preview of coming attractions. So a little teaser there, what happens when you do that. So you can stimulate a certain part of the brain and see what happens. Or you can go in and record the neural activity. You can record the electrical activity, chemical. You can go in and measure how many the neurotransmitters that are released. You can radioactively label them and see the activity. You can look at metabolic activity and cellular activity. So one way to do this is to look at the electrical signal, because action potentials, which we'll start talking about the next series of lectures, it's an electrochemical process. So an action potential, that's how information travels down a neuron. It's a change in voltage. You go from negatively charged to positively charged for a millisecond. And you can use microelectrodes or macroelectrodes to look at what's going on. So a microelectrode, you do this more in the lab. This has less clinical significance, but with a microelectrode, you can record the electrical activity of a single neuron. You can do it in humans, but usually this is mostly done in the lab. Just stick it in and just see that neuron firing. Macroelectrode, that's going to be EEG, electroencephalogram. And what's going on with the EEG is you put a series of electrodes on the scalp, and they gather the information and you get a brain wave. And you read the wave, and you can read different things. And I did this when I studied sleep. You can read brain waves and see if they're in REM sleep or deep sleep, if they're awake. Honestly, EEG is not a very good assessment tool. It's very broad. You're looking at large swaths of tissue to see general activity. And it's not as precise as we would like it to be. So if you want to know if somebody is, what their activity is after some accident, there was a recent case. A woman was in a car accident and she was declared to be in an unresponsive vegetative state. And they put her in an fMRI and told her to imagine she was playing tennis. And the proper brain areas for language and motion and visualization lit up. So there was something going on in there. Her brain responded to the command. So maybe she was just an active brain trapped in a skull, which is scary. So we get big ideas of what's going on. We could see some things. We could see big areas of tissue that are active or not active, but it doesn't mean we know what they're thinking. I think that technology is ahead of us. We'll get closer there. We're getting close to there already. But right now, EEG is not the most precise technology we have. Now, there's other technology we have that records the metabolic activity in the brain. So if neural activity increases in one part of the brain, the metabolic rate increases is the assumption. And what you do in one thing is you measure that with a radioactive 2-deoxyglucose, a radioactive dye. So it's similar to glucose, and glucose is taken up by active cells, active neurons in the brain. And so the assumption is that more active areas of the brain are going to take up this radioactive form of glucose, and then you could look at it and see, this is an active part of the brain. They must be doing X, Y, Z. And so one form of this is with a PET scan. PET scans stands for positron emission tomography. This was developed in the 1970s. Actually, one of my professors at UCLA got the Nobel Prize. Did he get the Nobel? Wait, I think they did. I know he developed it, so he developed this. And it gives information about the metabolic state of the patient, which parts of the brain are active. So this gives functional information, not just anatomical, not just, oh, look, we can see the structure of the brain, but what parts are active at any one time. So what you do is you inject this 2DG, this radioactive solution, and they emit positrons. And then you don't have to worry about the physics of it, but this radioactive glucose is absorbed by the most active brain cells, and it gives off a neutron and positron, and blah, blah, blah. Basically, gamma rays are going to be released, and you detect the gamma rays. And the computer is going to see which parts of the brain have taken up the most of this radioactive 2DG, and we assume it's the most metabolically active areas, and we get a picture based on that. And they're pretty pictures. All right, so an advantage to a PET scan is you see the activity to some degree, not just structure. So these are functional, not just anatomical. And they're pretty, which bonus points for that. But they are expensive. The half-life of 2DG, this radioactive substance we use, is really short. So the chemicals have to be produced on site in a cyclotron, an atomic particle accelerator. So the PET scans are expensive. And you do use radioactive material. It is safe. It's considered safe. You don't want to have more than one a year, but it is considered safe. And it's pretty slow. It can take minutes to do a single PET scan. And many hospitals don't have them, so it's not even an option to have this done. So why we use these? We use them in science, just in the lab to learn some things about the brain. We can get some idea of status in some mental illness and Alzheimer's, epilepsy. And so we can see which areas of the brain are active for different things. And so they're pretty. So they light up different colors of what's going on. So this is one of my favorites. This was a PET scan of somebody that was told to see different words. And the areas that brought up, this is going to be, so look at the scale here, this is going to be more active. The green and darker areas, green and blue, is less active. So when seeing words, these areas light up. This is the visual cortex. And over here, we're getting into reading areas of the brain. We'll do this in a few weeks. Hearing words lit up here. So this is temporal lobe. That's where you hear things. So you can see it's not real sharp and precise, but you can get a sense of more active, metabolically active areas of the brain and less metabolically active when doing certain tasks. All right, so those are PET scans. Last one I'll talk about is fMRI, functional MRI. And they modified the MRI. And so it gives both an anatomical, which MRI does, and a functional view of the brain. And fMRIs are hot, but we're starting to hesitate a little. So what these do is these detect changes in blood flow to particular areas of the brain. So you don't have to put the 2DG, the radioactive substance, in there. What they do is they measure oxyhemoglobin, which has a different magnetic resonance than deoxyhemoglobin. So a hemoglobin, and the substance in your blood, it carries oxygen. It has a different magnetic resonance than, so oxygenated areas different than deoxygenated areas, and so we can make assumptions about what's more active. And these are faster than PET scans. It's not radioactive. You do get a higher resolution. So everyone was like, hooray, fabulous. So you can see it's much sharper than we have than a PET scan, but we can see some activity as well. So advantages: they are faster, as I said, than PET scan, get a higher resolution. It's non-invasive, it's non-radioactive, but we are now starting to see some real disadvantages, and the main one is the accuracy. There is a high, what we call noise to signal ratio, or low signal to noise. You have to calibrate the machine precisely so you can get some response, but you're not just hearing random things. So if somebody's wiggling around, literally, that can skew your results. And so in one of my very favorite studies ever, what they did is they took a dead salmon and they put it in an fMRI. So here's the method section. You took a salmon. The salmon was not alive at the time of scanning. And they put a dead salmon in an fMRI and gave the dead salmon a task. The dead fish was shown a series of photographs depicting human individuals in social situations with specific emotional expressions. And the dead salmon was told to determine what emotion the photo was shown in the photo. And here's the results of the dead salmon brain. And you could say, woo! When dead salmon are analyzing human emotions in photos, that's the part of the dead salmon brain that analyzes it. Sputter, sputter. Clearly, that's a high noise to signal ratio. It's hard to calibrate these things. So clearly that's not a real finding. But so fMRI is not perfect. If the threshold is high enough so there wouldn't be any false positive, you wouldn't get any results. You have to calibrate it just right. And if it's set too low, then dead salmons think about human emotions. So you have to be real careful when you're interpreting this data. We also have to be careful with overgeneralizing. All right, the brain is really complex. So if you get somebody in an fMRI and you tell the person, your subject, to wiggle their finger and something lights up on the fMRI, is that area that lights up the only finger wiggling area? Okay, here's a better example. The area that lights up when you give to charity. You get somebody in and they do a thought experiment that they're giving to charity, and woo, a part of the brain lights up. Well, is that the charity region? Or is it, I don't know, guilt? Is it counting? Are they figuring out how much money? Is it fear? Are they thinking about what they're going to have for lunch? What does increased blood flow to an area mean? Don't confuse correlation and causation. Just because two things happen together, this area lights up in the dead fish doesn't mean that's the dead fish's area of analyzing human emotions and faces. And they found a lot of inconsistencies. So even though we said, look, here's the love area of the brain, and here's this area of the brain, after doing it once, what they did, and a big study came out recently, is they got people and they did it again. They retested some subjects. So they had them in and they did the fMRI and they looked at different things and they said, woo, this area of the brain lit up. And they got them in 4 months later and they had them do the same task. And there was little to no consistency. So it doesn't mean that this doesn't show different areas of the brain lighting up, but if you do something again, maybe you're interacting with it differently. You're thinking about what you're having for dinner instead of lunch. It's not a new activity anymore. And so there's a value in fMRI, but just be careful and don't assume that something lights up and that is the guilt area of the brain or that is the charity region of the brain. You have to design your experiment to have the optimal signal to noise ratio. You have to retest and you can't overgeneralize. And I'm just going to throw one more thing in here. It's not in your notes, but this is a hopeful new technology. I don't know if you've heard of the Human Connectome project. We'll talk about it later in the semester, but it's it's been going on for, I don't know, at least 10 years, multi-million-dollar project sponsored by NIH, National Institute of Health, and they want to map the human brain. They want to construct a map of both structural and functional neural connections in the brain. And what they did is they improved on MRI. So they took MRI and they improved the technology. It's even sharper than it was. It's better resolution. And they added some EEG to it, some behavioral techniques. They're scanning fraternal and identical twins to see any genetic similarities. So they have a better MRI, and they're throwing some other ways to look at behavior in there as well. So to close this out, think of what technology we had 100 years ago and think how far we've come and think how amazingly far we're going to come 100 years from now. So we keep learning and we use our brains to keep learning about how to learn about our brains. And I'll close it out with that thought.